Hello, everyone, and welcome to the Precision Medicine track on day 2 of the APJ Genetic Solution Tour 2020. My name is Ryan Harvey, and I'm your moderator for this session. This session will focus on the use of Axiom microarray technology for precision medicine applications, including population-scale genotyping, pharmacogenomics, and developing a polygenic risk score for Alzheimer's disease. Just as a note, if any questions arise during the following presentations, we encourage you to submit them in the Q&A box. Our speakers will address your questions following the presentations. First, we will hear from Shantanu Kushrikar, who is the Director for Product Marketing for Genotyping at Thermo Fisher Scientific. Shantanu is a part of the leadership team that manages our commercial and R&D efforts for our microarray business. Today, Shantanu will be providing you an overview about how predictive genomics is a powerful capability to help predict disease risk and understand drug response in order to improve health outcomes and manage costs. He will be talking about some of the large-scale population genotyping efforts implemented on the Applied Biosystems Axiom microarray platform, which have led to personalized health outcomes. Shantanu will also be speaking about how the Axiom microarray platform has been used to drive pharmacogenomics initiatives for clinical research. Welcome, Shantanu.
Thank you. The title of my presentation today is Predictive Genomics, Powering the Future of Population and Personalized Health. Predictive genomics is a powerful capability to predict disease risk and understand drug response in order to improve health outcomes and manage costs. Thermo Fisher is at the forefront of this wave of predictive genomics, where in the not-so-distant future, genomic testing brings with it a greater opportunity for managing disease and improving health outcomes. The accessibility of genomic content from diverse populations driven by government health systems and industry is accelerating the precise prediction and diagnosis of disease. Today, clinical care explains about 15% of overall health outcomes. Genetic data, which is relatively easy to measure, can add about 30%. And this, of course, is a huge opportunity for us to influence health outcomes. And what genetic data helps to do is stratify populations, target screenings, optimize treatments, and tailor therapeutics based on a person's biology. The UK Biobank study, where 500,000 samples were processed in the United Kingdom, was the first to take the step to integrate genomics to demonstrate that integrating genomic data can add value to understanding disease and stratify populations. The UK Biobank today has over 1,100 publications, the genetic data for which was generated on the Axiom genotyping platform. Today, over 75% of the requests to the UK Biobank are to access this genetic data so that researchers can conduct meta-analysis with newly generated data from the laboratory or to conduct their own research. Coupled with deep phenotyping rather than self-reported data, data from the Axiom genotyping platform has powered several revolutionary discoveries in common diseases as demonstrated by the publications from the Biobank. Techniques to identify genetic risk in populations such as polygenic risk scores are now becoming relevant because of the power of these studies, and bringing health and wellness related tests to the clinic is becoming a reality. The UK Biobank paper published in Nature demonstrated the utility of microarrays for large population screening initiatives and also showed how imputation-aware designs could be adopted for population-specific diseases. One of the outcomes of the UK Biobank has been to demonstrate the power of the polygenic risk scores. Various sequencing initiatives have attempted to catalog human genetic variation, for example, via the 1,000 Genomes Project. These initiatives have demonstrated that most of these variations will be entirely benign. Some may subtly impact on risk of various common diseases. Such variants with minor allele frequency greater than 1% have demonstrated that most people are at risk from some sort of common disease such as diabetes, obesity, cardiovascular disease, to name a few. To offer the benefit of genetics to the vast majority of the population, polygenic risk scores derived from genetic data can have predictive value for many common diseases. A very small number of genetic variants will have the potential to cause serious genetic disorders or disease, either in an individual or in their children, and are categorized as rare inherited disorders. While polygenic risk scores are not new, the UK Biobank demonstrated that with the Population Skill Initiative, polygenic risk scores can be developed that would offer the opportunity for asymptomatic, at-risk individuals to receive the appropriate management that might have otherwise not been considered, and also would reduce the amount of overdiagnosis in healthcare systems. The paradigm for the future then is to manage health and not disease. The early years of genotyping included several large studies, also on the Axiom platform, and included studies such as the Kaiser study that was processed at University of California in San Francisco. Over 100,000 samples were processed from this study. Other studies such as the UK Biobank with 500,000 samples and are continuing today are research-focused cohorts with the purpose of developing deep insights into the genetic cause of disease. These learnings are helping develop outcome-focused cohorts such as FinGen, such as those used by the Finnish Biobank, FinGen, which is a collaborative effort of the biobanks in Finland, and are sparking new national achievements in biomedicine and predictive genomic tests to help manage disease. Our collaboration with MedGenome, a personalized medicine company, has produced a population-optimized solution that can be used to develop consumer and predictive genomic tests relevant to the South Asian population. So it is necessary to develop polygenic risk scores for each population because the polygenic risk score is unique, and a single polygenic risk score cannot be applied across different population groups. This means developing a PRS for a population will require screening of several million people to help manage their health and to be able to benefit from genetics. Unlike any other whole genome technology, microarrays offer the capability to genotype both rare and common genotypes in several million people rapidly and cost-effectively. The Axiom platform offers the ability to customize arrays and is built from fully customizable modular genetic modules. These modules allow you to tailor an array for your research study. These modules include markers for pharmacogenomics, for blood groups and blood phenotypes, and disease-associated markers. Every Axiom array today includes a unique imputation-aware GWAS grid. That means that markers chosen on the array provide the most imputation power and enables us to impute the highest number of markers for one or more populations with the highest accuracy. Other modules within Axiom array include disease risk variants relevant to a wide range of diseases such as cancer, cardiovascular, and cardiometabolic disease, neurological disorders, Alzheimer's, Parkinson's, etc., to name a few. HLA typing is available for studying immunity and inflammation. And finally, pharmacogenomics for potentially adverse, for learning about potential adverse reactions or suboptimal dosing based on the latest genomic information. Every array is fully customized with content to match your project goals and offers high value, high utility content. The FinGen study that was mentioned earlier uses the Axiom platform with the goal of developing better ways to manage health for 1,100 disease endpoints. These include diseases in areas such as ophthalmology, neurology, cardiometabolic disease, rheumatology, to name a few. FinGen was genotyped about 10% of the population, which is about 500,000 people from Finland on Axiom arrays. By using imputation, they are able to accurately reconstruct the genomes of the Finns, which are then combined with the electronic health records from the National Health Registry. By performing association analysis and meta-analysis, the goal of FinGen is to move from discovery to treatment and prevention. Today, over 250,000 samples have been genotyped in about 2 years and have enabled discoveries and confirmation in diseases such as diabetes, inflammatory bowel disease, asthma, and macular degeneration. FinGen selected microarrays and chose Thermo Fisher Scientific because of our commitment to support their efforts and genotyping with microarrays, our ability to meet their goals of improving health outcomes in various disease endpoints within the lifetime of those that have participated in this study. The FinGen array is a fully customized array with an imputation-aware design and offers high genomic coverage in the finished populations. Thermo Fisher also offered analysis support and customized solution with the ability to redesign the array and add markers to the array every year. The modular diagram shown on the left slide shows the modular construction of the finished biobank array, which includes beyond the GWAS grid, other content such as pharmacogenomic content, disease-causing variants, immunity variants, and variants that are implicated in cardiac, cancer, autoimmune, and inflammatory diseases. Today, predictive genomics incorporates pharmacogenomic markers on every array. This includes both investigative biomarkers and valid biomarkers to learn about drug-drug and drug-gene interactions. Pharmacogenomics is integrated into our custom and catalog arrays in biobanking for population stratification and pharmacogenomics, in preemptive pharmacogenomics for clinical research and pharmaceutical drug trials. The Applied Biosystems Axiom PharmacoFocus and PharmacoScan arrays are economical way to generate focused pharmacogenomic data. The Axiom platform is the only platform that brings over 20 years of experience in the field of pharmacogenomics to address markers in pseudogenes and complex regions such as CYP2D6. The PharmacoFocus array that is shown over here is a low-cost solution that is available with over 2,000 relevant pharmacogenomic markers and integrated star allele calling. The array offers coverage of a wide range of therapeutic areas. Pharmacogenomic arrays such as PharmacoFocus include content that has been selected by consortium for their high evidence and deep coverage of critical genes that are relevant to pharmacogenomics. A unique assay, the Axiom genotyping assay, includes gene-specific amplification and makes possible the genotyping of complex genes or pseudogenes such as CYP2D6, CYP2C19, amongst others. The pharmacogenomic platform today is deployed at multiple sites. One such collaboration is with the University of Pittsburgh and the University of Pittsburgh Medical Center. Both UPitt and UPMC have deployed the platform to be able to look at hundreds of genes and to be able to offer preemptive pharmacogenomics to the 4 million or so patients that are today enrolled in the UPMC healthcare system. The goal of UPMC, as mentioned by Dr. Phil Empey, the Director of Pharmacogenomics, is to select the best medication for each patient based on their genes. And they have gone from one gene to hundreds of genes using the Axiom pharmacogenomic platform. The University of Pittsburgh, or UPMC system, is our center of excellence and aims to preemptively test 150,000 subjects in a reviewed research study and integrate the participants' pharmacogenomic profiles in the electronic health records to offer the right medication at the right dose faster. Starting with 4 million patients in the catchment area of the UPMC systems, over 150,000 patients will be recruited into their biobank with the goal of doing preemptive pharmacogenomics in a clinical workflow that includes over 14 genes and over 1,200, and a research workflow that includes 1,200 pharmacogenes that will form the basis of their translational research. The goal is to show the value of true population-scale preemptive pharmacogenomic testing by delivering innovation and best practices in the use of genomic data. This leads us to how will genomics be used in the future. As I mentioned in my first few slides, genetic data can add 30% more information to improve health outcomes. Polygenic risk scores and pharmacogenomics can then be combined to help stratify the risk in population and identify drug response, so that genetic testing can offer the opportunity for asymptomatic at-risk individuals to receive the appropriate management and the right drug at the right dose that otherwise might not have been considered. At Thermo Fisher Scientific, we continue to invest in various R&D initiatives to help introduce new whole genome capabilities cost effectively and rapidly so as to make an impact in improving health outcomes. This has included capabilities in pharmacogenomics, rare variant analysis, carrier screening, and more recently, the Axiom Propel workflow to be able to screen over 2 million samples per year. Our mission is to enable our customers to make the world a healthier, cleaner, and safer place. Thank you.
Thank you for the great presentation, Shantanu. We have several questions coming through now, and the first one we have here is, can pharmacogenomics further improve the initiative of drug repurposing in the treatment of COVID-19?
Thanks, Ryan. Yeah. So yes, pharmacogenomics can certainly improve the initiative of drug repurposing. And we recently launched an array very specifically for that, which is the Axiom Human Genotyping SARS-CoV-2 array, which has more than 6,000 pharmacogenomic markers. The idea really was to understand the drug-drug and drug-gene interactions and to determine whether any of the existing medications could be used for the purpose of repurposing, obviously in a research environment. And then the same module, which looks at the sort of the pharmacogenomic markers, also has variants to look at vaccine development and vaccine research. So certainly, pharmacogenomics can improve the initiative of drug repurposing. I hope this helps answers the question. And the array, as mentioned in the presentation, uses the gene-specific amplification step, which really looks at these critical genes.
Thank you, Shantanu. A couple other questions that have come through here. How does the Finnish Biobank design the genome coverage grid?
I'm sorry, could you repeat that again?
Yeah. How does the Finnish Biobank design the genome coverage grid?
Oh, so we did. So the good question that the Finnish Biobank design used an imputation-aware design. It basically it provided us with several markers, but we also leveraged the 1,000 genomes populations. And in addition to that, Thermo Fisher has a large Axiom database of validated markers. So these are verified markers. So we were actually able to design a Finnish-specific biobank array that had extremely high coverage and accuracy for the Finnish population, so compared to anything else that was available on the market.
Great, thank you. And I guess playing into that as well, what role does imputation-aware platforms play in these population-scale projects?
So imputation-aware designs are a great way to design arrays. So we typically call them population-focused arrays, and they are designed then through a process called imputation-aware process. And we can design them for one or more process. The unique design capability at Thermo Fisher allows customers to capture maximum number of common low-frequency and rare variants that occur in the population. And this is done with extremely high concordance and accuracy. So the arrays have higher performance, robust data quality for a variety of research applications, including pharmacogenomics, blood typing, where we are seeing increased contribution from low and sometimes rare alleles. So all in all, the population-specific arrays designed through this imputation-aware design process really help identify these important associations and allow for research into more disease endpoints. Does that help answer?
Yes, thank you, Shantanu. Another couple of questions we have here. Why are population genotyping projects such as the Tohoku Megabank, MadCap, UK Biobank, and FinnGen opting for microarray as the platform of choice? And what value do they see in creating custom arrays for these populations?
Again, another great question from the audience, Ryan. So the Tohoku Mega Biobank, as you may be familiar with, was designed in Japan by the Tohoku Mega Biobank to study the Japanese population. The MadCap array, if many people have not heard about it, was a cancer array for prostate and other cancers for African populations, and then of course the UK Biobank and FinnGen that I mentioned. The reason for designing these population-specific arrays was really twofold, and the population-specific arrays offer higher statistical power and the ability to conduct large studies, as mentioned in my presentations. And these are really to help identify these important disease associations and allow for research into more disease endpoints. A second reason for designing these population-specific arrays is with the goal of calculating polygenic risk scores for these populations. And we know that now the polygenic risk score is unique to a population. So by capturing the genetic diversity and effect sizes using variants that are relevant to the population in these studies, the arrays are able to overcome the Eurocentric biases, and they avoid interrogating sort of the monomorphic variants that are typically observed in generic sort of the general purpose catalog arrays, which are typically marketed as suitable for global screening. So really the goal is to make these important scientific discoveries and avoid missing out on important associations. That's the reason for designing population-specific arrays.
Great, thank you. And on a similar sort of note here, another question we have is, in the NGS era, why are these projects intended towards microarray data instead of RNA-seq or whole exome sequencing?
Can you repeat the last part of the question again, Ryan?
Yeah, so in the NGS era, why is the work that is being done here tending towards microarray, I guess, instead of next-gen sequencing?
Oh, yeah, a good question. So first thing, microarrays, it's the simplicity and ease of use as well as the cost. You can certainly run large numbers of studies. As I mentioned, we've already, the FinnGen has already done 250,000 samples in 2 years, already have started to publish papers on polygenic risk scores. But really it comes down to the data robustness. And in the age of NGS, there's certainly more discussions around whether low-pass sequencing can be used or just whole genome sequencing. Both of these, especially the low-pass sequencing, leads to quite a bit of missing data. It has challenges in data robustness. Depends a lot on imputation. So if you don't have your populations in your imputation panel, or if you don't have a good reference genome, which is very true for anything that is not European, then next-gen sequencing is certainly not attractive, and you want to be able to cover these studies very quickly. So microarrays are really the choice of technology for running these large-scale studies. And it provides this comprehensive solution, right? So you want to be able to look at pharmacogenomics. You want to be able to look at regions of HLA, so looking at immunity and inflammation, for example. You want to look at genes, very specific pathways, rare variants, which are increasingly having higher effect sizes in polygenic risk scores, for example. And so this makes the microarray platform uniquely suitable for these large-scale population studies. And that, really the being able to sort of screen large numbers of people within these countries is extremely important if you want to benefit from genetics.
Great, thank you. And a final question we have here as well is there any particular advantages or different or disadvantages between the Axiom platform and other competitors in the market in the microarray area?
Yeah, so good point again. There are 2 points really that I would like to call out. First one is I already mentioned earlier, which is the way we design these arrays is extremely unique. These are the imputation-aware designs that Thermo Fisher has a very specific bioinformatics capability that is not available on any other platform. The second is really around the robustness of the technology. And the way we manufacture the arrays allows for the batch sizes or the batches to be consistent, so there are no SNPs dropped across any particular batch. And this is extremely important when you're looking at variants in pharmacogenomics or even variants that could have high effect sizes that would have implications in how the disease is managed in the future. We've run ourselves examples here, and we've seen other platforms drop important markers just across a couple of thousand samples. So really, the robustness of the platform, the data that comes out, the coverage of the populations, and then more specifically, the markers and areas of the disease and traits such as pharmacogenomics is extremely important. This is one reason why customers have selected the Axiom platform for all of these large studies that I've mentioned here and more.
Great, thank you, Shantanu. And we have time for one last question here. Do you think in low resource, big population countries that these microarray solutions can be applied on a mass scale for screening and diagnosis?
Certainly, yes. And there is plenty of partnerships that are being we're working with very closely. MedGenome is a great example of a partnership in India, where they're looking at screening as many number of people as there are in India, so 700, 800 million. The idea here is really to do this cost effectively and in our lifetimes, right? So when we screen these populations, the goal is really to be able to extract the genetic information and make the decisions or changes in lifestyle based on the findings from our genetic profile. So really serving well from a large-scale perspective. And, this is the same thing is true for FinnGen. Also, Taiwan, which I didn't mention, is again, a good example where we have employed the platform across the country.
Great. Thank you very much, Shantanu, and thank you for the presentation and answering those questions as well.
Thank you, Ryan.
So next up, we have Dr. Kang Wong Hong, who is the Director, Division of Healthcare Innovation at Therogen Bio in South Korea. Therogen Bio have been conducting genomic research and offer genome-based testing services. Today, he will be sharing Therogen's experience in transitioning from the Applied Biosystems OpenArray platform to the Axiom microarray platform, and how Therogen partnered with Thermo Fisher to develop the Therogen PMRA chip for genetic testing services. Welcome, Dr. Hong.
반갑습니다. 방금 사회자께서 소개해 주신 테라제니텍스의 홍경원이라고 합니다. 오늘 제가 설명드릴 주제는 커스터마이즈드 테라젠 PMRA 칩 디자인 히스토리 and application examples라는 타이틀로 시작을 하겠습니다. 이런 주제를 선택하게 된 데는 테라젠이 유전자 검사 서비스를 하기 위해서 많은 부분에서 서머 피셔와 협업을 해왔고, 그 과정에서 가장 최적화된 그런 서비스 솔루션들을 만들어 왔습니다. 그러한 결과물들을 오늘 간략하게 소개해 드리고자 합니다. 오늘 제가 설명드릴 내용 콘텐츠는 총 4가지인데요. 먼저 테라젠 바이오가 가지고 있는 유전자 검사 서비스에 대해서 간단한 개발 과정을 설명드리고요. 그리고 테라젠이 이 대규모 데이터를 활용하기 위해서 PMRA 칩을 커스터마이징을 하였는데 그 결과에 대해서 설명드리겠습니다. 그리고 이런 PMRA 칩을 활용해서 어떻게 서비스를 활용하고 있는지와 또한 더 추가적인 연구에 어떻게 활용하고 있는지를 설명드리겠습니다. 테라젠 바이오의 지네틱 테스팅 서비스에 대한 내용입니다. 지네틱 테스팅 서비스라고 하는 것은 많은 사람들이 알다시피 어떤 개개인의 건강 상태에 대해서 영향을 미치는 요인들은 생활 습관적인 요인과 또 유전체의 요인 두 가지가 결합해서 나타나게 됩니다. 그래서 술을 너무 많이 먹거나 또 담배를 피거나 또 이제 과로를 하거나 하면은 우리 몸속에 있는 콜레스테롤이나 혈당 등이 불균형을 이루게 되고 그 결과로 만성 질환의 원인이 되게 됩니다. 이러할 때 유전적으로 어떤 부분이 취약한지 알고 그 부분을 고치게 되면은 좀 더 건강 상태가 개선될 수 있도록 할 수가 있고 그 결과로 질병의 위험을 예방할 수가 있습니다. 저희 테라젠의 서비스 목적은 개인이 갖고 있는 가장 취약한 유전적인 요소들을 확인해주고 그에 맞춰서 어떻게 건강 관리를 해야 되는지를 제시해주는 그런 서비스를 하는 것입니다. 테라젠이 이런 서비스 개발하기 위한 과정인데요. 먼저 첫 번째는 이런 서비스에 활용하는 유전자 마커를 어떻게 발굴하였는지입니다. 아마 각 국가마다 사용하는 마커들은 그 인종에 특화되어 있는 그 유전자를 쓰게 될 테고 또 그것이 보편적으로 많은 연구자들에게서 받아들여지는 그런 결과여야지만 이렇게 서비스에 활용할 수가 있을 것입니다. 저희는 그러한 부분을 지와스 카탈로그라고 하는 데이터베이스에서 1차적으로 질병이나 표현형과 관련되는 유전 지표들을 발굴하게 되고 그 이후 발굴된 유전 지표에 대해서 상세한 데이터를 S&P 페디아라고 하는 곳에서 분석을 하게 됩니다. 또한 마찬가지로 표현형과 관련돼서 다양한 크로스 밸리데이션한 스터디들을 휴게 내비게이트라는 곳에서 확인을 합니다. 이게 다양한 S&P 데이터베이스들을 활용해서 신뢰도 높은 마커를 발굴하게 되고 단순히 마커를 발굴했다고 해서 바로 쓰는 것이 아니고 임상적으로 혹은 고객에게 설명할 수 있는 그런 스토리를 만들어야 됩니다. 그런 스토리는 이 S&P와 연관되어 있는 유전자의 기능이라든지 S&P 자체의 기능 등이 중요하게 작용을 하게 됩니다. 그래서 S&P와 관련된 유전자의 기능은 징카드라든지 UCS 데이터베이스에서 확인을 하게 되고 S&P 자체의 기능은 GTX 포털이라든지 등을 통해서 S&P의 기능을 확인을 하게 됩니다. 그래서 기능적으로도 유전자의 기능적으로 S&P의 기능적으로도 어떤 표현형과의 설명이 명확히 가능할 적에 그 마커를 저희가 유전자 검사용 마커로 선정을 하게 되고 추가적으로 얼마나 많은 레퍼런스가 있는지를 기반으로 우선순위에 해당되는 S&P를 선정을 하게 됩니다. 그 최우선순위에 해당되는 S&P들을 고객들에게 검사용으로 사용하게 되고 그 결과 바탕으로 어떤 지네틱 리스크를 갖고 있는지를 설명을 하게 됩니다. 네, 이렇게 이제 찾아진 마커들을 실제 실험하기 위해서는 이런 실험할 수 있는 플랫폼을 디자인을 해야 되는데 처음에는 그 마커들의 프로브 위치들을 팩맨 기반으로 디자인을 하게 됩니다. 그리고 이제 이런 팩맨 실험을 싱글 어세이로 하기보다는 대규모 샘플 또 대규모 마커를 한 번에 실험하기 위해서 오픈어레이라는 플랫폼을 사용하게 됩니다. 오픈어레이라는 플랫폼에는 32개부터 64개, 128개까지 배수로 이제 늘어나는 마커들을 한 번에 검사를 할 수가 있습니다. 그 마커들을 디자인한 칩을 저희가 구매를 하게 되면 4주에서 8주 사이에 테라젠 도착을 하게 되는데, 그때 이 칩이 제대로 워킹을 하는지 각 프로브별로 이 지노타입 간의 이 구별이 명확하게 될 수 있는지를 밸리데이션 하게 되고, 1차 QC를 거친 마커들만 가지고 저희가 서비스에 활용을 하게 됩니다. 물론 여기서 QC를 페일 하게 된 마커들은 저희가 오픈어레이 칩을 업그레이드 할 적에 그 마커 이외에 LDA 관계에 있는 또 코릴레이션 높은 주변에 있는 다른 마커들을 활용해서 보완하는 그런 방향으로 마커를 옵티마이제이션 하게 되고, 그 결과로 저희가 이제 한 10년 정도 이런 서비스를 하면서 이 오픈어레이 칩에 있는 프로브들은 가장 최적화된 형태로 지금 구성되어 있습니다. 그 최적화됐다는 것은 실험할 때마다 그 지노타입 결과들이 일정하게, 또 동일한 샘플을 실험했을 때도 동일한 결과가 일정하게 나올 수 있도록 세팅이 완료된 상태에서 레귤러한 서비스를 진행을 하게 됩니다. 그리고 이제 실험 결과가 나오게 되면 실험 결과는 저희가 서버에 업로드를 하게 되는데, 저희가 갖고 있는 서버를 RIMS라고 얘기를 합니다. 실험실 자동화 시스템이라고 하는데, 여기에서는 실험 결과를 자동으로 분석을 해서 결과 리포트를 만들게 됩니다. 그런 분석 과정에서는 그 마커들이 갖고 있는 위험도 정도를 계산을 하게 되고, 그런 것을 지네틱 리스크 스코어라고 얘기를 합니다. 지네틱 리스크 스코어를 계산하게 되면 그 스코어를 위험도 수준에 따라서 총 4단계 혹은 3단계로 나누게 되고, 위험도가 높은지 낮은지를 결과지에 출력을 하게 됩니다. 물론 그 결과지 자체가 오타가 있는지, 또 어떤 페이지가 누락이 있는지 등을 추가적으로 체크를 하게 되고, 최종 고객에게 납품을 하게 됩니다. 이런 식으로 개발된 칩들이 저희는 2010년부터 해서 계속해서 업그레이드를 해 왔었고요. 저희는 2010년에 헬로진이라고 한 서비스를 처음 출시한 이후에 매년 새로운 헬로진 버전도, 최근에는 진스타일 다이렉트라고 해서 한국 내에서 가능해진 BTC 유전자 검사 서비스를 이 오픈어레이 칩으로 디자인을 해서 서비스를 하고 있습니다. 하지만 이제 이런 오픈어레이 칩만으로는 다양한 고객들의 니즈를 모두 만족할 수가 없었고, 또한 중요한 포인트는 유전자 검사 혹은 유전자 연구를 통해서 새롭게 발굴되는 유전 지표들이 매년 기하급수적으로 많이 늘어나게 됩니다. 그래서 그렇게 늘어나는 유전 지표들을 오픈어레이를 매번 디자인하면서 다 반영할 수 없기 때문에 우리가 그런 것들을 선제적으로 대응하기 위해서 PMRA 칩을 개발을 하게 되었습니다. 이 PMRA 칩은 원래 이제 썸오피스에서 아시아 PMRA 칩으로 이미 커머셜라이징 되어 있는 그런 칩인데, 이 칩을 저희가 약 5만 개 정도의 마커를 추가적으로 커스터마이징을 하면서 테라젠 PMRA 칩으로 개발을 하였습니다. 물론 이렇게 이제 PMRA 칩으로 서비스를 하더라도 대규모 수주라든지 대규모 서비스권이 있을 적에는 PMRA 칩으로는 어떤 TAT라든지 또 가격 조건 등을 맞추기가 힘들기 때문에 특정한 경우에는 오픈어레이 칩으로 서비스를 하기도 합니다. 예를 들어 이제 아모레에서 최근 출시한 아이오페 티모 인덱스 같은 경우에는 한 번에 2만 개의 샘플을 실험해야 되는 케이스가 생겨서, 이런 경우에는 이제 PMRA 칩으로 실험하기에는 너무 한계가 있기 때문에 오픈어레이 칩으로 서비스를 개발하고 서비스 진행을 하고 있습니다. 이와 같이 이제 오픈어레이 서비스를 오랫동안 해왔고, 지금도 물론 다양한 고객들에게 판매를 하고 있지만은 몇 가지 이제 리미테이션들이 있습니다. 오픈어레이 칩은 저희가 이 결과지 혹은 결과 리포트를 만들기 위해서는 선제적으로 준비해야 되는 시험 준비 과정이 굉장히 길게 걸리게 되고 또한 그 디벨롭 기간이 오래 걸립니다. 또한 이제 한 번에 검사할 수 있는 마커의 개수가 한정적이다 보니 최근에 발굴되고 있는 신규 마커들 모두 커버할 수가 없는 그런 단점이 있었습니다. 그래서 이런 부분들을 테라젠 PMR의 칩으로 모두 다 커버할 수 있게 개발을 하였습니다. 다만 이제 PMR의 칩 또한 몇 가지 리미테이션이 있는데 TAT, 샘플을 결과까지 만드는 데 걸리는 시간이 아무래도 오픈 어레이 실험보다는 좀 더 길게 걸릴 수밖에 없습니다. 또한 조금 더 코스트가 높은 그런 리미테이션은 있습니다. 하지만 이런 리미테이션에도 불구하고 이 칩이 갖고 있는 유연성, 즉 다양한 연구 결과물들을 반영을 빠르게 할 수 있고 또한 서비스뿐만 아니라 많은 연구자들의 니즈를 다 맞출 수 있는 그런 칩이기 때문에 저희는 이제 이런 칩으로 서비스 플랫폼을 변환시키고 있습니다. 이런 테라젠 PMR의 칩을 개발하는 과정에 어떻게 디자인이 되었는지 간단히 설명을 드리겠습니다. 이 테라젠 PMR의 칩은 원래는 아시아 PMR의 칩 혹은 그냥 PMR의 칩이 기반이 되어서 개발이 되었는데 저희가 PMR의 칩 혹은 이걸 엑시옴 기반의 칩이라고 얘기를 합니다. 엑시옴 기반의 칩을 선정을 하게 된 데는 아무래도 타 경쟁사의 칩에 비해서도 좀 더 드랍 레이트가 낮고 좀 더 정확도가 높다는 판단하여서 진행을 하게 되었습니다. 이런 PMR의 칩은 다양한 분야에서 적용이 가능한데 무엇보다도 이제 숨겨져 있는 칩들이 또 단순히 이제 지놈 커버리지가 높은 마커들만 숨겨져 있는 것이 아니고 기능적으로 중요한 유전 지표라든지 또 임상적으로 중요한 유전 지표들 또 이 지놈 커버리지를 높이기 위한 임퓨테이션을 높이기 위한 마커들도 많이 속해 있고 또한 그 파퓰레이션에 특이적으로 발견되는 로우 프리퀀시 마커들도 많이 포함되어 있습니다. 그래서 레어 베리언트 연구를 하거나 질병과 관련되는 진단용의 연구를 하거나 또는 생물학적인 기능의 연구를 하거나 또 유전체 커버리지를 높여서 홀 지놈 시퀀스에 대비되는 어떤 결과물을 만들고 싶을 적에 PMR의 칩으로 많은 것들을 할 수가 있습니다. 그래서 이런 PMR의 칩을 선택을 하였고 여기에 물론 이제 이 PMR의 칩에 아시아 PMR의 칩이 됐을 적에는 그 앞에 PMR의 칩보다도 이스트 아시안 파퓰레이션의 마커들을 더 많이 포함할 수 있고 또한 아시아 파퓰레이션에서 발견된 지네틱 베리언트들이 더 많이 포함되어 있기 때문에 한국인이나 동아시아인뿐만 아니라 동남아시아인이나 중동아시아인까지도 다 커버할 수 있는 그런 칩 콘텐츠라고 생각이 되었습니다. 그래서 이 칩에 테라젠이 지난 10년간 연구를 하면서 발견한 약 5만 개의 SNP를 추가로 커스터마이징 하면서 저희가 테라젠 PMR 칩으로 개발하게 되었고 그 결과물로서 기존에 이제 아시아 PMR에 침겨져 있는 칩 마커들 이외에도 DTC 유전자 검사 마커라든지 한국인 특이적인 마커를 더 추가하고 또한 테라젠이 그동안 연구해왔던 R&D 결과물들을 추가를 하여서 총 5만 개의 마커를 더 추가를 하였습니다. 그 결과로 82K에 해당되는 SNP를 검사할 수 있는 칩으로 개발이 되었습니다. 오픈어레이 칩에서 테라젠 PMR의 칩으로 넘어가면서 다양한 이제 장비적인 부분도 변화가 있긴 하지만은 기본적으로 마커의 개수가 80만 개 이상 한 번에 검사할 수 있는 장점이 되었고 또한 저희가 이제 이 서비스를 하면서 이 TAT를 굉장히 단축시켰는데 2주 내에도 충분히 서비스할 수 있는 것으로 확인이 되었습니다. 또한 이제 저희 자체적으로 데이터셋들을 테스트해 본 결과 콜레이시라든지 디큐시아 같은 PMR의 칩의 퀄리티 자체가 굉장히 흡수하게 나오는 것을 확인했고 저희가 연구용뿐만 아니라 서비스용으로도 이상이 가능할 수 있었습니다. 물론 이제 이렇게 되기까지는 초반에는 이제 콜렛이 많이 떨어지는 그런 샘플들도 많았습니다. 그래서 이 테이블에 보시는 것처럼 초기 약 5, 6개월 정도는 저희가 로우 콜레이트를 가지는 결과물들을 사실 실험률이 높았었는데 저희가 이제 그 라이브러리라든지 그 파일들을 실험 결과를 분석하는 그 로우 데이터 분석 프로그램들을 업그레이드를 하면서 대부분의 샘플들이 95% 이상의 콜레이트를 보였고 99% 이상의 샘플이 굉장히 많이 늘어나는 결과를 가져왔습니다. 그래서 그 결과로 지금은 이제 안정적으로 연구든 서비스 목적으로 다 활용이 가능이 됐습니다. 이와 관련된 어떤 칩의 스페시피케이션은 기존 아시아 PMR의 칩의 스페시피케이션은 거의 대부분 다른다고 볼 수 있습니다. 그래서 지놈의 액큐러시, 칩의 액큐러시 같은 경우는 94%, 95% 이상을 가질 수 있고 또 이스타 엔세스트리 지놈의 커버리지는 약 99.7% 이상 가질 수가 있습니다. 그래서 이렇게 이제 개발된 테라젠 PMR 칩을 이용해서 이제 여러 가지 서비스를 하고 있는데 테라젠은 이제 한국에서 2020년부터 시작된 DTC 시범사업 확장판을 국내 서비스를 하고 있습니다. 그래서 총 55가지의 검사 항목을 서비스를 하고 있고 여기에 사용되는 유전자가 약 400개 이상의 유전 지표들을 한 번에 검사를 하고 있습니다. 이 400개 이상을 검사하기 위해서는 당연히 테라젠 PMR 칩이라는 것을 활용을 해서 검사를 하고 있고요. 그리고 이제 이 강점은 한 번에 400개 이상 또 55가지 이상 항목을 검사를 한 이후에 어떤 추가적으로 고객의 요구사항에 따라서 피부 혹은 건강관리, 영양소 등 별개의 검사 항목 카테고리로 분리된 결과물들을 추가로 서비스할 수 있음으로써 부가적인 이 매출 효과를 올릴 수가 있었습니다. 또한 이제 제가 칩을 개발하면서 가장 이제 중점을 두었던 것이 개인의 유전체 데이터를 저장을 해 두었다가 개인이 필요할 때에 어떤 관련되는 결과물들을 받을 수 있는 시스템을 개발하는 게 목표였고 그런 것을 테라젠에서는 진뱅킹이라고 하는 서비스로 해서 준비를 하였습니다. 그래서 유전 테라젠 PMR 칩으로 실험한 결과물을 테라젠의 서버에서 보관을 하고 있다가 고객의 요구에 따라서 고객의 결과물을 추가적으로 혹은 최근의 연구 결과물들이 더 늘어남에 따라서 개인의 결과가 업그레이드 된 경우에도 고객에게 그런 업그레이드 결과물을 추가로 제공해 줄 수 있도록 진뱅킹 서비스를 진행을 하고 있습니다. 이에 더불어서 이제 병원에서 의사분들이 가장 원하는 것이 이 영양소를 어떻게 처방을 할 거고 또 이제 다이어트에서 어떤 부분을 중점적으로 다이어트 집중을 높일 것인지 이런 부분 고민을 하게 되는데 그런 곳에 이제 참고 지표가 될 수 있는 검사 서비스들도 가지고 있습니다. 그래서 뉴트리션이라고 해서 바이타민이나 또 미네랄과 같은 영양소들이 부족해질 수 있는지 이런 것들 검사를 해서 그게 맞춤형으로 영양 관리를 할 수 있도록 이렇게 서비스가 개발되어 있고 메디핏이라고 해서 비만이라고 다 같은 비만이 아니고 어떤 당대사, 지방대사 등 어떤 대사 특성에 따라서 비만의 성향이 달라질 수 있기 때문에 그런 대사 특성들을 구분해서 분석할 수 있는 메디핏 또한 개발을 하였습니다. 그래서 이러한 검사들은 기본적으로 100개 이상의 마커, 많게는 약 500개의 마커를 가지고 검사를 하고 있기 때문에 이런 경우에도 테라젠 PMR 칩으로 서비스를 하고 있습니다. 이렇게 예시로 이제 메디핏 같은 경우는 비만이더라도 단순히 이제 그냥 비만 위험도만 검사하는 게 아니고 이게 스트레스 때문인지 지방 대사 때문인지 에너지 소모가 안 되는 건지 당 대사가 안 되는 건지 등을 검사를 해서 그 위험도가 높은 부분에 대한 다이어트 관리를 해주는 서비스를 하고 있습니다. 이런 이제 PMR 칩 기반의 서비스 이외에도 다양한 리서치들을 하고 있는데요. 저희가 제주도나 아모레 퍼시픽과 함께 맞춤형 화장품에 대한 연구를 수행하고 있고 이런 이제 신규 유전 지표들을 테라진 PMR의 칩 기반으로 발굴을 하고 있습니다. 예를 들어 이제 지금 이 그림에서는 피부에 어떤 트러블이 생기는 부분이 그 비누를 바꿨을 때나 또 화장품을 바꿨을 때에 트러블이 잘 생길 수 있는지 아닌지에 대한 그런 결과를 바탕으로 테라진 PMR 칩을 이용한 지와스 분석을 하였고 그 결과로 이제 어떤 히알루로닉산에 관련되는 유전자를 발굴하게 됐습니다. 이와 같이 이제 테라진 칩은 굉장히 촘촘하게 마커들이 숨겨져 있고 또 펑셔널 베리언트들이 많아서 지와스 분석을 했을 적에 의미 있는 베리언트를 찾기가 아주 용이한 것으로 확인이 되고 또한 이제 다양한 클리니컬 인덱스들에 대한 지와스 분석을 해서 기존에 다른 칩으로 나왔던 또 한국인 칩으로 나왔던 결과들이 충분히 리플리케이션 되고 검증되는 것을 확인을 할 수 있습니다. 이제 이런 칩 기반으로 결과들을 모바일 헬스케어 서비스로 개발을 하여서 개인의 라이프로그 데이터 혹은 개인의 건강검진 데이터를 결합해서 건강관리를 매일 모니터링할 수 있는 시스템으로 활용도 하고 있고요. 또한 이제 뭐 얼굴의 변화에 대한 지와스 연구를 하여서 얼굴 각 부위별로 연관성이 높은 유전자를 발굴한 연구도 하고 있습니다. 또한 이제 이런 데이터를 활용해서 계통 분류적인 분석을 했을 적에 어떤 그룹 간의 차이를 분명하게 나뉘는 그런 S&P 조합을 찾을 수 있고 그걸 바탕으로 어떤 그룹의 분류를 정확하게 할 수 있었습니다. 여기 나와 있는 그래프는 팔로제닉 트리는 체질과 관련된 것입니다. 체질에 따라서 이렇게 주황색 체질을 가지는 사람과 파란색 체질을 가진 사람이 저희 칩을 이용하여서 팔로제닉 트리를 그렸을 때 정확하게 구분이 되는 것을 확인할 수 있고 이게 기존의 이제 체질의 어떤 생물학적인 기반이 부족한데 그런 것들을 이제 보완해주는 그런 에비던스로 활용이 되고 있습니다. 그래서 저희 결과물들은 뭐 다양한 지와스 연구뿐만 아니라 임상 속에서 퀘스천을 가지고 있는 그런 부분들 해소하는 데도 활용이 되고 있고 또한 추가적으로 제가 계획하고 있는 그런 콜라보레이션들이 20가지가 넘는 연구들이 진행이 되고 있습니다. 그리고 거기에 대해서 이제 이 칩에 심겨져 있는 약물 유전체 마커들을 이용한 약물 유전체 연구, 또 CMV를 분석할 수 있는 PMR 칩의 강점을 이용해서 CMV를 이용한 그 연구, 그리고 칩에 심겨져 있는 유전성 암 마커들을 브라카와 같은 그런 유전성 암 마커들을 검사할 수 있는 연구, 또 희귀 질환의 돌연변이를 보유하고 있는 분들을 검사할 수 있는 희귀 질환 돌연변이 고인자 검사를 연구를 하고 있습니다. 이런 식으로 이제 PMR 칩이 활용될 수 있는 분야가 굉장히 다양하고 지금까지 이제 짧지만은 저희가 그동안 경험해왔던 부분들을 컨사이스하게 설명을 드렸습니다. 혹시 뭐 질문사항 있으면 질문을 남겨주시고 발표는 여기서 마치겠습니다. 감사합니다.
Thank you for the great presentation, Dr. Hong. We have a number of questions coming through now from the audience. And first up, how are turnaround times of OpenArray service and microarray service different from sample to report?
That is a good question. And during the seminar, OpenArray format was introduced. We usually use 64 format OpenArray, and the experiment starts when we have at least 48 samples. So overall, OpenArray service requires maximum 2 weeks including sample collecting time. But however, for microarray, the experiment starts when we have at least 96 samples. So it takes longer time to collect the samples than OpenArray service. Therefore, for this reason, microarray service needs minimum 2 weeks.
Thank you for that. Another question we have here. Phylogenetic analysis was very interesting. Are you providing the genetic testing service by using phylogenetic analysis data?
We are conducting the research collaboration for phylogenetic analysis, and with the result of this research, we will intellectual property rights, and then we are planning to provide the genetic testing service. Maybe in next year, yes.
Great, thank you. I think call rate can be affected by diverse factors such as sample, population characters, and so on. What do you especially focus on for increasing call rate up to 99%? Is it a Korean population specific SNP set?
It is the important question, and actually there was a small effect of sample or population character. Call rate was increased by using upgrade library file for genotype cluster. So with the origin method of genotype cluster, the sample which is out of calling area shows no core. So thus we optimized core area in order to decrease the rate of no core.
Thank you. And we have time for one more question here. Medifit Nutrition Service, Medifit and Nutrition Service look good to manage health and eating habits. Do you provide food allergy information as well?
I'm sorry, I missed the last part of your question, so could you tell me again?
Yeah, not a problem. The question was, Medifit and Nutrition Services look great to manage health and eating habits. Do you also provide food allergy information as well?
Yeah, there are lots of the data from abroad, but we don't have enough local data about food allergies. So therefore, it is possible to provide overseas service, but not for Korea.
Great. Thank you very much for that, Dr. Hong. If you did have a question there that wasn't answered, we will reply to you by email and answer your question as well. So our last speaker for the day is Dr. Richard Pither from Japan. Dr. Pither is the CEO of Cytox, and the Cytox group of companies provide non-invasive risk assessment and patient stratification tools for Alzheimer's disease and dementia. Dr. Pither will be speaking to us about the utility of polygenic risk scores for identifying individual genetic risk and disease risk prediction in Alzheimer's disease at both the early and pre-symptomatic stages of the disease. Welcome, Dr. Pither.
Hello, my name is Richard Pither. I'm the CEO of Cytox, and I'm speaking today about polygenic risk score and the potential in Alzheimer's disease for clinical trial design and the optimization of clinical trial design. I'd like to give a little bit of an introduction to Alzheimer's disease and then talk more generally about polygenic risk scores and how they can be used. specifically in the context of Alzheimer's disease genetics and data that we published recently in the longitudinal analysis of one of the very important Alzheimer study cohorts, and what this the implications for this type of approach to the clinical trial enrichment and clinical risk management for Alzheimer's disease patients ultimately. So Alzheimer's disease today affects over 46 million people worldwide, and this figure is set to double by 2030, largely due to the aging population around the world. The current techniques are highly invasive, very expensive, and actually have relatively poor accuracy. And we'll talk a bit more about those techniques later. Cytox is developing a polygenic risk score that we call GENOScore, and this is a genomic test for assessing future risk of developing Alzheimer's disease, and in particular the cognitive decline associated with that disease. It's a relatively simple test requiring genomic DNA, which can come from either blood or saliva. We extract genomic DNA, run this on a proprietary genotyping array that we call Variatect, which is a bespoke array built for us by Thermo Fisher on the Axiom platform. And that array is read in the GeneTitan instrument, and basically 500,000 data points emanate from that analysis. That primary genotyping data is then read by our SNPfitter software, and that generates the GENOScore report for each subject. And we analyze essentially 96 samples with each run. And we've worked around the world with partners in the US in the form of Akesogen, in Europe with Eurofins, and in Australia with the Ramachandran Centre. So this is a robust platform that can be used with any laboratory partner that runs that GeneTitan Axiom platform. Now, late-onset Alzheimer's disease represents the majority of Alzheimer's disease, something like 98%, and it has a very complex etiology. In this illustration here, we can see that effectively the genetic risk for Alzheimer's disease is an inherited risk, and it's fixed throughout your lifetime. But whether or not someone develops Alzheimer's disease is a function of the environment and the lifestyle risk factors that individual encounters, as well as their genetics. So we can see here that cardiovascular risk, diet, exercise, the presence of diabetes, neuroinflammatory processes can all affect the time and the extent to which the cognitive decline and other symptoms associated with Alzheimer's disease become manifest. And we know from identical genetic twin studies that these environmental factors have a major impact on when or whether an individual is likely to succumb to Alzheimer's disease. So in other words, genetics can explain part of the risk, but management of lifestyle and environmental factors and risks are very important for Alzheimer's disease clinical management as well. On this slide, we can see that some of the biomarkers in use today have really changed quite profoundly the way we think about Alzheimer's disease. On the left-hand side of this chart, on the graphic, we can see that individuals start declining with late-onset Alzheimer's disease symptoms from about 60 or 65 years onwards, but it can be very unclear whether or not that cognitive change, those symptoms, are due to normal healthy aging, or whether there's underlying Alzheimer's pathology developing in the brain in the form of the amyloid plaque and the neurofibrillary tangles and the neuritic plaques. We know now from imaging and histopathological studies that this pathology can actually start maybe 20 or 30 years before clinical symptoms. So that provides a fantastic opportunity, a window within which therapies may be administered to prevent the ultimate manifestation of Alzheimer's disease. Now, the way that we look for these pathologies today can be very invasive and very expensive. So amyloid imaging using positron emission tomography, or PET, has taken off really in the last 5 to 10 years and really transformed our understanding, but is really of limited routine clinical use. The other option is to look for the presence of amyloid and tau fragments in the cerebrospinal fluid using lumbar puncture, and this obviously carries its own risks and has seen so far limited clinical application. We work at Cytox very closely with Professor John Hardy, a world-renowned geneticist, and he's of the view, as we are, that genetic analysis is basically the way to provide the most cost-effective and reliable stratification of subjects at highest risk. Now, Alzheimer's disease genetics is complex. It involves the presence of DNA variants across the genome. By far the most important genetic variant identified so far is that associated with the ApoE gene, apolipoprotein E. But we can see on this Manhattan plot that there are a fairly large number, more than 40 variants across the genome that reach genome-wide significance level, either in the context of risk association or indeed protection for late-onset Alzheimer's disease. So we need to take all these into account when we're bringing together a polygenic risk score. So we've worked closely with a number of collaborators around the world, and indeed with the team at Thermo Fisher, to put together 500,000 markers, all of which have a degree of association with risk or protection for Alzheimer's disease. So you can see we've divided on this graphic the content of our array into various different panels, including work that's emanated from the group at UCL, University of Birmingham, the so-called mTOR panel, the very important Cardiff University panel, which we'll talk about in a moment, and then some other content from non-Alzheimer related neurological conditions, as well as the early onset dominant inherited Alzheimer mutations as well. So it's a very comprehensive array. We believe it's the most comprehensive genetic array developed so far for Alzheimer's disease risk association. Now, I mentioned the Cardiff University content on that array, and that's very important. We've worked very closely over a few years with Professors Valentina Escott-Price and Julie Williams and their teams at Cardiff, because we believe they have the most effective polygenic risk score algorithm so far developed for Alzheimer's disease. You can see some of the characteristics here. The algorithm was trained on the IGAP, so-called IGAP consortium data, which involved 17,000 Alzheimer cases and 37 controls. And in the paper referenced here, Valentina basically published 87,500 SNPs, which best described Alzheimer's risk. In fact, we've moved this work on now with Valentina and her team, and we used 114,000 SNPs from the array in our analysis. And you can see here that the work published initially with Valentina showed an 84% accuracy in predicting Alzheimer's disease in one particular cohort in this paper, but there's been subsequent validation of this work in several other cohorts as well. Now, very importantly, there are a number of polygenic risk score algorithms that have been published from different academic groups around the world. And in 2018, there was a review of the relative merits and performance of each of those algorithms. And it's very clear from this paper, authored by Hannah Stocker and colleagues, that the Cardiff University polygenic risk score algorithm, which is the basis of our Cytox GenaScore polygenic risk approach, was the best performing. And therefore, that's the reason that we chose that as the basis for our commercial product development. Now, a little bit about, before we go into some data, a little bit about Alzheimer genetics. We mentioned the ApoE, the apolipoprotein E gene earlier, and it comes in several genetic forms, predominantly E2, E3, and E4. And of course, every individual carries 2 copies of that gene. We can see on the right-hand side here that about 25% of the population carry 1 or more copies of the E4 allele, and E4 has an Alzheimer risk association. Certainly, if you carry 2 copies, you're at elevated risk. If you carry a single copy, you're considered to be at higher risk than average, but there are lots of people who carry E4 who will never go on to develop Alzheimer's disease. So it's important that any polygenic risk score consideration includes an E4 component, but they're also important that we can also differentiate between high and low risk even amongst the E4 carriers. Conversely, the bulk of the population don't carry E4 at all. So we can see here in this publication from the UK that about 60% of individuals carry the E3 homozygote profile, and that they're considered to be at moderate level of risk. And yet we know that about 40–45% of Alzheimer's sufferers come from this non-E4 carrying E3 homozygote group. So it's important that we can stratify for risk amongst those individuals. Just to add to the complexity, if we carry a copy of E2, then E2 is a relatively low risk allele for Alzheimer's disease. So if you were an E4, E2 heterozygote, you would carry a lower than normal risk for Alzheimer's disease development. It's complex genetics. So what are the benefits potentially of being able to assess individuals for Alzheimer's disease using this polygenic risk score approach? And I've listed several of those opportunities here, but there are others, of course. I mean, the obvious one is by identifying clinical trial participants that are most likely to decline cognitively within the context or the time frame of that clinical trial. We're likely to be able to use reduced subject numbers and therefore lower cost approaches to clinical trial recruitment, thereby increasing the chances of demonstrating a therapeutic effect, which tends to be done using cognitive test measures. We have to show that the advantage of a polygenic risk score over and above just using ApoE genotyping alone for the reasons I just described. And of course, what we're really interested is understanding whether we can predict those at highest risk of cognitive decline, not just for clinical trial enrichment, but also for optimizing clinical care ultimately. A final advantage of this type of approach is that you can begin to understand genetic profile that may link to specific molecular pathways, which would allow you potentially to identify those most likely to benefit or indeed not benefit from one particular therapeutic intervention. So a real personalized medicine opportunity. So we've done much of our analysis for GenoScore in the so-called ADNI dataset. ADNI is the Alzheimer's Disease Neuroimaging Initiative. And this is a longitudinal study that's still running today. It's been running for over 15 years, headed by Professor Mike Weiner at the University of California, San Francisco. And what we've done is used data from about 630 subjects confirmed with using amyloid imaging to contain amyloid in the brain, so they would be considered to be at higher risk of Alzheimer's disease. And then we've used also amyloid negative controls. So we've got well-matched age groups. We've got an ApoE genotype prevalence that would be normal for Caucasian cases and controls. And we've used this very rich data set to analyze the performance of our genetic polygenic risk score approach. So a little bit of data now on the next couple of slides. So what we can see here, if we just concentrate on the highlighted area, the red highlighted area, we can see that if we use ApoE alone across this ADNI cohort, we have an AUC of about 73%. If we use the PRS alone without the ApoE component, we're at 70%. If we combine the two together, we get to about 80%, which is a very good level of accuracy in describing biomarker-confirmed Alzheimer's cases from age-matched biomarker-negative controls. So that's very encouraging. What we'd really like to know is whether we can stratify the subjects in the E4 or indeed the non-E4 carrier group. So what we did here on this page is we looked at the distribution of polygenic risk scores in the E3, E4 heterozygotes. So these would be considered just by ApoE genotype alone to be at relatively high risk. But we can see on the right-hand side that there's a distribution of polygenic risk scores all the way from 0 to 1, 1 being the highest. And if we apply a cutoff, a polygenic risk score cutoff here of about 0.6, we can see a positive predictive value of around 84% in identifying genuine AD carriers at this, or AD risk subjects within this universally high-risk cohort. Conversely, if we look at the E3 homozygotes who would be considered to be at low risk of Alzheimer's disease, and again here we apply a cutoff at the to remove the lowest quartile, we have a negative predictive value of about 89%. Whereas if you apply a highest quartile cutoff, you improve to about 51% the positive predictive value. So you can see here the opportunity to enrich for the highest and lowest risk individuals, regardless of their ApoE4 status. And this has not been possible genetically before now. Now, as I said earlier, the real prize here is to be able to predict those individuals at the very early stages of cognitive decline who are most likely to decline further over the course of a clinical trial period, usually up to 4 years. So here what we've done is taken baseline individuals with so-called MCI, or mild cognitive impairment, and then we've asked how many of those people have changed significantly using a cognitive rating scale called ADAS-Cog over the next 4 years, and how accurately could we have predicted those at highest risk of cognitive decline. So you can see here of the 285 MCI baseline individuals, 61 cases declined by 10 points or more over a 4-year period, which is considered clinically significant, whereas 224 remained stable, and that we were able to identify with an AUC, an accuracy of about 74%, those at highest risk of cognitive decline just using a polygenic risk approach. This is highly interesting data. And then if we look at that group slightly more closely on the left-hand side here, you can see that the distribution of ADAS-Cog decline mimics very nicely, or matches very nicely, the polygenic risk score. So on the x-axis, we can see polygenic risk ranging from 0 to 1, and we can see that those with the highest degree of decline, 15 points, are bunched up towards the right-hand end of this graphic, the highest polygenic risk scores. The lowest of 5 points or fewer are distributed mainly to the left, as you would have expected, and those who decline by 5 to 15 points, intermediate level, again have a nice rightward shift in this distribution, showing this link between polygenic risk score and cognitive decline over a 4-year period. If we look on the right-hand side of this graphic, we can see that the ApoE4 homozygotes, the people coloured in red, are at the highest end of the polygenic risk score spectrum, as we would have expected. Those that carry one copy of E4 in yellow are distributed towards the right-hand side, but you can see there are plenty of relatively lower risk individuals that carry E4. And the E3 homozygotes, again, distributed from very low polygenic risk to much higher polygenic risk, again illustrating the fact that you're able to identify those at highest risk of developing the disease amongst the E3 homozygotes. So, in conclusion, we have been able to show that we can stratify for E4 carriers at the highest risk of developing Alzheimer's disease, and conversely, we can also stratify the E3 homozygotes at the highest and lowest end of the risk spectrum for developing Alzheimer's disease. And this is really of critical importance both to pharma companies looking to stratify clinical trial populations, and of course also clinicians who want to understand risk in their subjects. The algorithm has the potential to improve the ability to predict those with early mild cognitive impairment who are at highest risk of developing Alzheimer's disease, and this is an improvement over using ApoE genetics alone. And of course this would allow you to select subjects at the highest risk and the most likely to decline in the context of a clinical trial timeframe, thereby allowing a very valuable enrichment opportunity. I mentioned also that we have an approach looking at pathway analysis, and importantly, our GENOScore algorithm we are launching as a clear LDT assay in the US later this year, and as a CE marked product in Europe also by the end of this year. And this is really important because the last 20 years has been very unsuccessful in identifying new therapies for Alzheimer's treatment, but there's some very promising new drugs coming forward now. At the front of that pack is aducanumab, being developed by Biogen. Biogen presented some very interesting new analysis of their clinical studies at the end of last year, and they're currently in discussion with the FDA about resubmitting this for approval during the second half of 2020, with the expectation that there may be some kind of approval a year or 18 months after that. So the opportunity for treatment and improved treatment for patients is becoming more of a reality. So it's important that testing keeps pace with that. Finally, I would like to thank my colleagues in Cytox for much of the work and the analysis that I presented today, and also pay tribute to our scientific partners, particularly at the Dementia Research Institutes at Cardiff University, the teams led by Valentina Escott-Price and Julie Williams, the UCL team led by Professor John Hardy, and also the University of Exeter team led by Professor Clive Ballard. I'd also like, of course, to acknowledge ADNI for access to their data, and colleagues at Thermo Fisher who've provided a lot of technical guidance over the course of this work. And finally, as well as our investors in Cytox, we've also received funding from Innovate UK here in Oxford, and also from the European Union through the Horizon 2020 research program. Thank you for your attention.
I would like to thank Dr. Pither for his presentation today, and I do need to correct that Dr. Pither and Cytox are UK-based rather than in Japan, as I previously stated, so my apologies for that mix-up. Dr. Pither wasn't able to join us live today, but he has prepared some answers to questions that had come up earlier. And if you have any additional questions from that presentation, please feel free to send these through, and we will do our best to answer them now or pass these along to Dr. Pither as well for his attention. What do you think the practical limits of polygenic risk scores for risk stratification will be in routine use?
So as I mentioned earlier, the Alzheimer's disease has both a genetic and an environmental and lifestyle risk component. We've done much work to understand the limits of polygenic risk scores for late-onset Alzheimer's disease, and we believe that the polygenic risk score algorithm, which has an upper limit of accuracy of around 85%, is performing at that level now. So we think that the polygenic risk approach that we've adopted effectively takes into account all the genetic variation that will be contributing to risk in an individual. But of course, the time and the severity of the symptoms in any one individual will also be impacted by lifestyle and environmental risk exposure over the course of an individual's lifetime.
How has your polygenic risk score been validated? Has there been any independent verifications?
So our polygenic risk score algorithm has been validated in multiple data sets. The Cardiff University group on which our algorithm is based has been validated in a number of longitudinal, very well characterized samples, 3 or 4 independent verification data sets, including individuals who went on to die, and then we were able to look at post-mortem histopathology confirmation in Alzheimer cases and age-matched controls. And that's a sort of gold standard approach to validating polygenic risk score algorithms. And in fact, the performance of the algorithm in that particular study was at something like 86 or 87%, which we think is at the theoretical maximum for this type of polygenic risk score approach.
How do you design your Axiom array to make it as effective as possible for polygenic risk score analysis in AD?
So we've designed the Axiom array that we call VariaTect to contain the ideal SNP combinations to run the polygenic risk score, the genus score analysis. That, as I mentioned in my presentation, includes 114,000 SNPs or DNA variants that are optimized for the Cardiff algorithm or genus score approach. And then we have a lot of other content which allows us to do other forms of analysis, including identifying individuals with the early onset high risk DNA variants or mutations in presenilin 1, presenilin 2, and amyloid precursor protein, which we use as a quality control step to make sure that we're not dealing with an early onset case as we do our analyses. We also have DNA variants associated with other non-Alzheimer's disease dementias on the array. Again, that allows us to broaden the type of analysis we can do in any one data set.
Who are the key potential customers for your polygenic risk score?
Potential customers for our PRS approach today include both academics and pharma companies. Pharma companies obviously are driven by the need to stratify the individuals at highest risk into their clinical studies so that they can optimize cohort selection for demonstrating therapeutic effects of their particular interventions, be they pharmacological, i.e. new drugs, or other types of interventions. So we've been working with a number of those companies over the last couple of years. Also, academics are interested in understanding genetic and non-genetic factors for Alzheimer's disease risk. And so we've worked with various academics to help them understand genetic risk profiles within their own cohort, their own longitudinal subject cohorts.
Do you envision your array and software going through a 510K regulatory approval?
So our array and software, which together comprise the GenoTOR product, we've been in discussion with the FDA in the US for a few weeks. We've decided, with their advice, that we will pursue initially the approval and launch of a CLIA LDT or laboratory developed test service in the US with various lab partners. And in Europe, we will go down the CE marked route, and that allows us to get this product available to a very high quality standard by the end of this year for the US and Europe. And but beyond that, we will be doing the formal clinical validation studies that would support a 510K regulatory process and approval in the US and in Europe. And we would imagine or envisage starting those studies later this year, and then being in a position to file for regulatory approval sometime during 2021.
And that's the end of the live Q&A for Dr. Pither's presentation. I really want to thank all of our speakers for their time and efforts today, and for everyone listening for the questions as well. This concludes the webinar sessions for today, but the environment will remain live and open, so please drop by our booth and speak to us and find out more about the solutions that we have to help accelerate your research. Please also participate in the scavenger hunt game. Look out for our icons in the environment to win some cool prizes. There will be a pop-up survey appearing in a minute. Please complete this to help us evaluate this roadshow, and thank you again for all of your attendance.